National Institute of Neurology in Mexico, we will talk about the updates of mechanism of protection and the neuronal damage of neuroglycopenia. The brain requires a high, the continuous supply of energy. It occupies 25% of serum glucose, which is equivalent to approximately 5 to 10 grams of glucose per hour, or 140 grams per day of average. In fact, the gray matter of the human brain uses 10 times more glucose than any other organs in the body. It is an important point for the complication of hypoglycemia. We will talk about this later. Glucose reaching from the 54 milligrams per deciliter in prolonged fasting to 200 milligrams per deciliter after lunch. Definition is serum glucose value of fewer than 50 milligrams per deciliter consistent with neuroglycopenia symptoms, or serum glucose value of fewer than 40 milligrams per deciliter without any symptoms. The mortality rate is between 4 to 10%, and this attributable to several hypoglycemia. The etiology is very varied. The most common are hypoglycemic drugs, and another etiologies are prolonged fasting or exercise, iatrogenic tumors, systemic infections, kidney or liver diseases, and alcoholism. So making the diagnosis is very simple due to the symptoms of hypoglycemia, which are present in the with mild, moderate, or severe. In mild and moderate symptoms, they are also called sympathoadrenal or neurogenic due to the activation of the autonomic nervous system by releasing epinephrine and norepinephrine that consisted on hunger, diaphoresis, tremor, dry mouth, palpitation, anxiety, eye tingling, although it may also be changed in condition attentional type. If left untreated and glucose continues to drop, it begins the neuroglycopenic symptoms of blurred vision, confusion, headache, dizziness, irritability, bradycardia, drowsiness, bradycekia, and fatigue that becomes very severe with seizures, coma, of death. It is important to mention that disorder is always present in the same way, because the occasions that have happened to him and the time is which that he has been in hypoglycemia are involved, because I have present as hypoglycemia encephalopathy, which is the main differential diagnosis of stroke, not only due to loss of motor or sensory functions, but also mainly due to the decrease in the performance of cognitive tasks, such as verbal fluency, reaction time, arithmetic skills, verbal and visual memory. This is demonstrated in 1990 by Ryan, evaluating the cognitive effects after a hypoglycemia event in children. He observed a significant decrease in the test of child-making attention and decision-making in the mild hypoglycemic group. In addition, was the value of blood glucose, but no recovery was observed in attention or reaction in the time test. After the symptom, the regularly very clear, a serum or capillary glucose measurement is performed, showing that the patient has less than 50 milligrams per deciliter. On the other hand, this is not much of diagnostic imaging in this patient, and other isolate events with rapid recognition and resolution. Although there are case reports, for example, if the patient was of 63 years old patient with hypoglycemia detect the time of onset of symptom now, and on complete approach, but performed showing this Brian MRI with hypersignaling sequence of T2, flare, and diffusion with restriction and ADC maps in the cortical and subcortical gray matter without undefined vascular territory. Despite the fact that in the managed and compatible with the stroke, the possibility was ruled out. Therefore, do not forget that in patients with symptoms compatible with the stroke, perform a glucose study per hand, otherwise the differential diagnostic can be complicated. The mechanism of the disease lesion is not clear, There are some theories that weed out at the bottom most acceptable if the first one. The first theory is that neuronal damage due to the toxicity. Second is cerebral ischemia due to the vasospasms and or vascular occlusion secondary to hemodynamic and hemostatic changes produced by hemoconcentration, hypercoagulability, or cardiac rhythm. Disturbance secondary to vegetative change and occur within the brain hypoglycemia. And the treatment is logical. This is only the application of glucose. However, some scenarios must be taken into account in the classic things is that if a patient is diagnosed with hypoglycemia, they are treated with glucose solution at 50%, which is equivalent to 25 grams of glucose. But currently, it's not recommended as it has been documented that applying the treatment is this way can resolve the symptoms without complication or recap complications due to the significant osmolarity of the solution. That there are high rates of exacerbation from 10 to 30%. On the contrary, excess variable glucose, which leads to increased corruptions in the tissues to repletion and surprises glucogenogenesis and glucogenolysis. This is in the end can lead to rebound of hypoglycemia. Therefore, survive for the last 3 hours in the emergency department is also recommended. Therefore, the infusion of glucose solution at 10% is currently recommended until the symptoms resolve for the last 30 to 60 minutes. Take into account that is patient with kidney or heart failure, a 20% glucose solution should be used to beat a different metabolism in this patient. With this new strategy, the complications that I have already mentioned are prevented and at a acute resolution of the symptoms achieved. Even some monitoring of the couple more hours is important because ATP concentration remains low in the brain even 3 hours after glucose concentration are restored. Often, instead of physiology, glucose is ingested orally passes the gastrointestinal tract to then enter the microcirculation to sodium-dependent glucose transport or SGLT, the one, and glucose transporter or GLUT2 receptors in epithelial cells. To the travel through the bloodstream until in the reaches the blood-brain barrier, and enter the central nervous system through transportation process by SGLT and GLUT1, which is found in the blood-brain barrier, and GLUT3 in the neurons. The happenings is the following 4 steps. First, the close veins to the carrier protein to the another phase of the cell membrane. Then, the carrier protein change conformation and glucose enters the cell membrane. It releases the glucose into the cytoplasm, and finally, the transportation returns to this is original reconfirmation, and this glucose beading sign is the esteroid. Again, in astrocyte, glucose can be used or undergo a transformation to the glycogen for the stores, while the neuron is used by entering the Krebs cycle to obtain 30 to 31 ATP molecules per each glucose molecule. In this figure, we can see the blood-brain barrier of the cell showing the fusion of the mitochondria and the change in the electrolytes and glucose. However, the part that I want to present is that when there is a greater requirement for glucose, the glutaminergic activity simulates the entry of glucose anthracytes, which also metabolizes stored glycogen as well as lactate. Since each molecule of lactate produces 70 molecules of ATP. With the happiness when we are in the situation of hypoglycemia and we do not have enough glucose to maintain brain metabolism. There are many glucose sensors neurons spread among the body, including peripheral glucose sensing neurons, which are located mainly in the carotid body and the hepatic portal mesenteric veins, as well as brain glucose sensors that are widely spread both, mostly found in the hypothalamus and the hindbrain, mainly in the area postrema and the dorsal and motor glucose and the vagus, and the nucleus of the solitary tract, where the cell's activity obeisance mechanisms that we will review in the next slide. When these mechanisms fail, mainly when there is decrease for a lower of 18 milligrams per deciliter of glucose. There is in electrical signs silence due to the cessation of cerebral electrical activity. This is because one of the main chains is the ionic overload of the cytochromes and in the mitochondria due to the sodium-dependent receptors. Initial a loss of polarity of the cell's membrane, causing ATP synthesis of to act differently by metabolizing ATP instead of production it. As neural diffusion continues, there is greater loss to membrane polarity in an initiation the release of excitatory amino acids, such as aspartate and glutamate. We are expressed from excitatory receptors such as NMDA and AMPA, thereby increasing the flow of intracellular calcium. This accumulation of calcium promotes lipolysis, increasing the amount of free fatty acids due to the metabolism of cellular membrane phospholipids, promoting release of reactive oxygen species, as well as causing platelet aggregation and neutrophil chemotaxis, leading to cell inflammation due to the direct damage and indirectly, finally, by reproductive factors such as cytochrome C, caspase 3, and apoptosis-inducing factor with unrelated, thus ending in the cell death. Therefore, we can conduce that hypoglycemia constitutes of mechanism of metabolic brain injury, and this will depend on the time that the patient remains of this hypoglycemia states. It is now that neural death begins after 50 minutes and that the damage is irreversible, with greater susceptibility in the cerebral cortex, hippocampus, and striatum. Therefore, if there are repetitive episodes of hypoglycemia occurring, one of the main damage will be the cognitive decline, thus as we mentioned in the previous slides, irreversible. Of course, one of the most complex systems with the have multiple regularity mechanisms in the nervous system. Hypoglycemia is no exception. We are going to the activity when glucose drops to less than 70 milligrams per deciliter with decreasing insulin production. When glucose drops to the less than 66 milligrams per deciliter, growth hormones and cortical are release of hepatic glycogenolysis and glucogenesis, as well as adipose tissues ketogenesis and lipolysis. Use of glucose by another peripheral tissue, such as muscle is increased. If glucose continues to below 54 milligrams per deciliter, there will be glucagon secretion followed by an increase in adrenaline to the further increase the above mentioned mechanisms. The starting points are important because that's been that in that of hypoglycemia occurrence, it can use the loss of these regulatory mechanisms and create an ambitious cycle that increase a risk of severe hypoglycemia with each event, with autonomic regulatory mechanisms are lost. Patients are able to recognize some sympathomimetic and neuroglycopenia symptoms, thus increasing the risk of severe hypoglycemia with a high possibility of causing coma, irreversible brain damage, and death. This is called hypoglycemia unawareness or happy hypoglycemia. As a regulatory mechanism located in the central nervous system, this is important to mention that the brain does not decrease in the metabolism due to the hypoglycemia. So the first localized mechanism in raising cerebral blood flow. The brain can use alternative to glucose, such as glycogen, and another is of is these substrates, such as lactate, pyruvate, and ketone bodies that can last from 2 to a maximum of 5 minutes before being depleted. The cellular level experimental models have shown that repeated events cause overexpression of GLUT1 and GLUT3. However, it is also counterproductive because the clinical manifestation can be unsighted and leads to hypoglycemia unawareness. Another now intracellular mechanisms is in the anthracytes is they can release proteins made from adenine, especially adenosine and guanosine, leading to cell repair after brain injury. Finally, calcium accumulation can also activate regulatory mechanisms to maintain adequate levels of this ion, such as calciquestrin and chelation promoted by the endoplasmic reticulum and mitochondria. So, areas to investigate the time which leads to actual long-term brain injury. Although the methodology will be very difficult due to the time and the number of events needed to discover great neuroprotective mechanism, how to restoration, self-regulatory mechanism that have been lost, and treatment in cognition impaired patients. In conclusion, glucose is the main fuel for the proper function of the central nervous system. The main mechanism of entry and use glucose at the molecular and cellular levels in the brain, emphasizing the neurons and astrocytes interact to form a common metabolic pathway, generating and protect effects to avoid an hypoglycemia coma or a major brain injury that leads to the cell death. We cannot forget that when a patient will already have neuroglycopenia secondary to hypoglycemia, they already have a change in that their metabolism on the recurrence becomes more frequent with each episode, which is why some insulin depend in diabetic diet. Is all. Any question?